First things first, we have to figure out what are essential amino acids and non-essential amino acids. Okay, I've given you the names here, but it's important to know the structures of these amino acids. Okay, for example, just some like very regular things. You see methionine, right? Methionine is the first codon and the first amino acid to be coded always, right? It has a sulfur group, right? Another one that has sulfur group is cysteine, right? Um, some of them might be acidic in nature, some of them might be basic in nature, right? Depending on what type of side chains they possess, right? Some of them are very charged, versus some of them are very neutral. Okay, so it's important to keep some of these basic amino acid structures in mind and special points about them, because they will definitely come in handy in certain questions when we look at them. Okay, now you see, as I said, so the two out of the 20 amino acids that you may know, there's only two amino acids which are actually acidic. Okay, aspartic acid or aspartate, or glutamic acid or glutamate. Okay, it's acid when it's in just acid nature. If it's a salt, then you call it aspartate with the ate in suffix. Okay, out of those 20, only three, or yeah, only three are basic. Okay, histidine, lysine, and arginine, right? So they probably have the extra NH2 group, and or they have like a couple of electropositive rings, etc. Okay, but at least we have to know about this. For example, they might bring it to you in what are histones composed of, right? They're very heavily positive, electropositive amino acids, right, that bind to negatively charged DNA. So they might ask you these things. So we have to know what type of amino acids and what special characteristics they have. Okay. Next thing is talking about gluconeogenic versus ketogenic amino acids. Okay. So you see, purely ketogenic amino acids are just leucine and lysine. Okay. Both of them, and as you see, lysine is basic. Okay. Leucine right here. Okay, that's the structure. And then lysine, as you see, that heavy NH2 group. Okay, so these are the only two ketogenic amino acids. Okay, everything else is a mix of it. So what I would do to remember it, remember the ketogenic and remember the glucogenic, right? Everything else can be in the middle, right? So glucogenic, we're going to remember methionine, histidine, and valine. Okay, methionine, histidine, and valine, right? So if you remember these two, you will remember, or at least process of elimination, the other amino acids out. Okay, so that's what we are sort of working with. Now let's move forward. I've given you another diagram that can help you understand a lot of these gluconeogenic versus ketogenic amino acids, right? So you see the red ones are all glucogenic, right? And the green ones are just the ketogenic. Okay, so try to remember, you see they're trying to enter, and why do we, or when do we need these glucogenic amino acids? It's when you're having gluconeogenesis, right? When your body is starved, you finish probably your glycogen stores, you're finished, or you're consuming your fats, you're consuming your proteins. That's when these guys are going to be entering the TCA cycle. Okay, so some of them enter as alpha ketoglutarate, some of them might enter as succinyl-CoA. Is it important to remember how are they entering? No, but a couple of these reactions are important. For example, the relation with glutamate to alpha ketoglutarate, okay, or alanine to pyruvate, right? These are called transamination reactions, right? So it's very important to remember these two amino acids and their reaction. Okay, apart from that, you might not see too many of these, but we'll definitely cover what's important. Okay, again, acidic versus basic amino acids, and you see why the acidics are acidic is because if, let's say, this is the principal carbon with the COH and the amine group attached, they have an extra carboxyl group, right? Both of these, right? Whereas the histidine groups have an extra NH, right, or a ring, or the NH3 side chain attached to it. Okay, now we're going to cover purines, metabolism, and all of that when we do molecular biology, but I want to start to integrate a lot of this amino acid and their derivatives into what we learned today. Okay, so as you see, purine formation, right, adenine guanine formation happens because of three amino acids. Okay, Aspartate, right, which is the acidic one. Glutamine, right, it's glutamate derivative, right, and then glycine, which is the simplest amino acid of all, right. If you look at the structure of glycine, it's just the typical, you don't have any side chain, it's only hydrogen. Okay, so one simple side chain of glycine, right, glutamine contributes the two nitrogens, then aspartate. Okay, whereas pyrimidine is just all aspartate. Okay, so it's very important to remember, right, how important aspartate is just to DNA formation. Okay, now I hope you're starting to correlate a lot of these things, and when we do look at them, we'll look at them thoroughly. Okay, now there's some chemical concepts we have to know, to not confuse ourselves when they give you these terminologies. Okay, because amino acids have both acidic group and basic group, right? They exist in two forms, right? They can exist in the acidic form, right, where you see low pH ends up charging the basic side of things, right? Or they can exist in high pH, which basically means that now the acidic side of things is charged. Okay, so they can be a positive ion or they can be a negative ion, depending on the environment they are in, right? So this concept is called pKa. Okay, now if the pH is lower than the pKa, it means the group is protonated, right? Which basically means it's in the acidic form, right? So you see protons, right? Positive, positive here, right? Which means pH of this ion or this amino acid, sorry, right, is lower than the environment that it's in. Okay. Whereas, the opposite goes where, you have the basic medium and now you see the deprotonated version. Okay. Now, why are we, usually doing this is because this pKa of this side chain helps me to understand how acidic something actually is or how basic something actually is, right? You see, arginine, the pKa of arginine is 12.48, which means it's very high, right? 7 is neutral. And now you see that it's a basic type, right? As you go down, and you see glutamic acid and aspartic acid again having that low pKa, right? Which means they're acidic, right? pKa high, basic, pKa low, acidic, right? So that's what this means, right? So I hope the terminologies will make sense. There's, I can elaborate it on in a little bit more. Usually we do that in farm, but for now, this is enough. Okay. All right. Let's keep moving forward. Another relational concept that they like to ask, and one we will look at in molecular biology, is translation, right? When you're translating, right, you use this universal codon table, right? So we need to learn how to read it. So if they ever give it to you, you at least know how to read the table, okay? You're not confused with it, you know what's going on, right? Now we know codons are made up of three nucleotides, right? Now you see the first letter is on one margin, the second letter is on in the horizontal margin, and the third letter on this margin. Okay. Now you start by reading the first, right? Let's say I'm making methionine, right? A, I go into the column U, and then I go into the row that is the G, right here. Okay. AUG is methionine, right? So AUG is the start codon, AUG is the methionine, is gonna show up as methionine, right? Now, do you see methionine written anywhere else? No. Right? So methionine is only one codon and only one amino acid, right? Now, why, where does it get confusing, right? There's a thing called wobble hypothesis, which basically means, and I'll go down here, right? You see, this was the mRNA, right? This is the anticodon loop, which recognizes the mRNA, right? It has the opposite of what the mRNA has, right? And you see, with the wobble hypothesis, what it basically says is this bonding is stronger than this bonding, right? Which is stronger than this bonding. So wobble means that this one is very wobbly, right? And because it's wobbly, it may recognize, C as G. Sometimes it may recognize C as, A or U or something, right? And because of that hypothesis, you see there might be overlap between the codons and the amino acids. For example, see CU produces leucine, right? CUC also produces leucine, right? And then you come here, UUA produces leucine, right? all of these are producing leucine. Why? Because there might be some sort of, affinity problem, right? And it's not necessarily a problem, but affinity issue that may mimic other amino acids. Okay, that's just about it. They're not going to ask any questions about it, but for you to understand how, why there's overlap between these amino acids, why there's four, codons coding for glycine and glycine only, right? Whereas aspartate, you see aspartate is right here, right? There's only two. Okay, so it is important to remember at least the main ones. So AUG you have to remember, then UAA and UAG you have to remember, yeah, and last one UGA, okay, because all three are stop codons. Okay, so if you go on and when we do mutations, frameshift mutations, and all of that, we've just built the base of understanding better molecular biology. Okay, so this is the thing, and then eventually we'll go into the structure, right? I have another example over here. You see, here is the 5' untranslated region. It finally recognizes AUG, so it gives you the methionine. It goes and you take, understands AUC, right? And if you see AUC, so A, U, and C, right? Um, isoleucine, I think, right? So isoleucine is here, right? Then eventually it recognizes UCG, which is serine, right? And then it recognizes stop codon, UAA. So you made a 3-chain amino acid, right? Polypeptide. Okay. Now, once these polypeptides are formed, they undergo a lot of bonding, right? They have a lot of hydrogen. They might undergo hydrogen bonds, right? They have a lot of, electropositive side chains. They might go, some sort of electrochemical bond, right? So if they form peptide bonds and peptide bonds only, then it's amino acids, right? Simplest one, insulin, right? It's a bunch of amino acids that, maybe mature into the final structure. Alpha helix, right? Alpha helix just means it's a helical structure, okay? Usually stabilized by hydrogen bonds. Tertiary structures, as you see here, they orient in a certain pattern, and they usually may, make folds, right? They may make up these enzymes. These are usually enzymes, how they fold, right? They might have disulfide bridges between them, right? And as I said, the most important amino acid, right, which have sulfur in their composition, it's methionine and cysteine, right? And if I just show you quickly, right, if I just go back up here, you look at methionine, right? The sulfur is also like bound, right, by the methyl group. When you look at the cysteine structure, right, cysteine is the only one that can actually make disulfide bonds, right? So whenever there's a disulfide bond that they're talking about, it's usually cysteine that they're discussing. Okay, quaternary structure, this looks like hemoglobin, right? Bunch of subunits coming together and making that one large hemoglobin structure. Okay, all right, now getting into the GI aspects of things, because we do need to discuss all of these, right? Now you see protein digestion and absorption, right, starts at the stomach. Okay, stomach gastric acid is very important, right? Denatures the ingested protein, activates pepsinogen, right? Pepsinogen converts into pepsin and starts cleaving all these peptide bonds. Okay, now once you start cleaving this, you're not actually done any major digestion or like absorption, right? You're just cleaving long chain into medium chain amino acid structures. Okay, where the main thing actually happens is the main organ that's responsible is pancreas, right? Pancreas meet the food bolus in the duodenum. And duodenum, how does duodenum activate these enzymes, right? Pancreatic and intestinal proteases cleave the peptide chains, eventually lead to oligopeptides, right? They might start absorbing some of these. There's two types of peptidases, right? One that break the chain from the inside, and one that cut snips of the amino acid chain one by one from outside, right? So there's endopeptidases and then there's exopeptidases. Okay, endo are trypsin, chymotrypsin, elastase, and that's about it. Okay, trypsin, chymotrypsin, elastase. Okay? Exo is carboxypeptidases, amino, or maybe dipeptide bases. Okay, so out of all of these, what we're gonna remember, trypsin, right? Chymotrypsin, elastase, and exopeptidases, only these. Okay, that's the only one that's typically important. Okay, the thing is, if you make trypsin, right, or if you make trypsinogen and you spill it, the activation of trypsinogen is dependent on enteropeptidase, right? This is a enzyme made by, and I'll show you right here. You see, trypsinogen is dependent on enteropeptidase, as it means entero, meaning intestines, right? Now, once this intestinal enzyme acts upon trypsinogen, only then can the trypsin be actually working, right? But in some cases, whenever they talk about acute pancreatitis and stuff like that, trypsin activates within the pancreatic duct, right? And it leads to autodigestion, right? So that's a clinical correlate. But typically, what's supposed to happen is trypsinogen is supposed to meet this enzyme, is supposed to eventually make trypsin, okay? And that endopeptidase continues to cleave all these bonds, right? Arginine, lysine. They've given you specific amino acids that they like to cut at, okay? Chymotrypsin cutting at this. And now once trypsin is activated, now it just catalyzes everything, right? It goes and catalyzes even the exopeptidases. Okay. So do we need to remember all the different sort of amino acids where they cut? No. But do we need to remember the enzymes and how they're activated? Yes. Okay. Now I've given you a couple of pages of these high yield correlations in first aid. These are all in the GI section of the first aid, 2025 or 26. And you see all the pancreatic secretions, especially focusing on proteases and even trypsinogen, right? How they're activated by enterokinase/enteropeptidase. It's the same thing. And what they actually work at. Okay. Last thing I've mentioned here, you see, they ask you all these different parietal cells. What do parietal cells make? What do chief cells make? What type of hormones are they using, right? And I want you to start looking at these, right? If you haven't seen it in a while, start reviewing them, okay? You see pancreatic cells, right? You see CCK can activate pancreatic cells, right? And then the ducts can release all of this and, lead to activation and stuff, right? Secretin activating the pancreatic ducts, okay? GIP made by K cells, that's more of like a carbohydrate thing, stuff like that, but still, right? So start looking over these high yield sections in the GI physiology. Okay. Now, finally, when you sort of destroy everything, you want to absorb it. So how do you absorb it? You have a sodium channel, sodium amino acid co-transport that takes in amino acids. Okay, so are you going to absorb anything without the salt? No. You need some amount of sodium to absorb that salt. Okay, and eventually it's just facilitated diffusion that happens because of transporters, and it goes into your blood vessels, right? Amino acids can travel throughout the body, but eventually a portal vein, everything will go to the liver, right? I've also given you important, this page with all the hormones that are required. You see, fatty acids, of course, fatty acids, and then you see all the proteins that are involved too. Okay. All right. Lastly, just looking at all the gastrointestinal secretory products. So gastric cells, they do like to ask the histology of these things, right? I would recommend Googling a lot of histological sections of these gastric pits and then recognizing which cells are which, right? So you see gastric cells, gastric acid is made by parietal cells, right? Then eventually leading to, that protein digestion that we talked about, right? Autoimmune destruction of the parietal cells will lead to eosinophilic histology, chronic gastritis, and pernicious anemia. You may see lymphocytes in that histological aspect too, right? And as long as we're here, you see these are the parietal cells, right? Imagine if T lymphocytes were located on just this region, right? It should be apparent that this is meant to be shown as autoimmune gastritis. Okay. Usually on top of the gastric pit is mucus cells. In the middle is the acid secreting cells. And down below is enterochromaffin cells that basically help in formation of certain neurohormones, right? Gastric hormones and stuff. Okay. So do review this again. Chief cells making the pepsin with the protein digestion, and then Brunner glands and other things that secrete bicarbonate into the matrix. Okay. All right, now moving forward. Now, what I recognized is over the years, people get confused with a lot of terminologies. Okay. The thing is, if you have made amino acids, you can have amino acids being made internally by your entroplasmic reticulum and Golgi apparatus and packed and sent out of the cell, right? Um, so you can do that, right? But there are specialized products, specialized functions that you can perform by combining those amino acids with carbohydrate structures. Okay. Now, why do we need to perform all this? We'll go into the functions of them, right? But there are three main groups that you have to start to, correlate together, right? Be able to discern them apart, right? Out of these three, similar, confusing looking substances, right? I want you to focus on proteoglycans, right? Proteoglycans, right? You see, proteo is the amino part, glycans is the carb part, right? Very high amounts of carbohydrates are found in proteoglycans. Okay. Now, proteoglycans have a special type of carbohydrate, which is called glycosaminoglycans, right? So this thing right here is a part of proteoglycans, not to be confused with anything, right? I've put this here because the terms look similar and people get confused in them, right? GAGs make up proteoglycans, right? You see carb type, long linear GAG chains. Okay. Core protein, we'll talk about this. Location is usually extracellular matrix, right? So just to give you a quick example, right? A lot of our joints have this colloidal type of fluid, right? Joint fluid, right? It holds up a lot of water, so it can dissipate weight better, right? So what are those proteins slash proteoglycans that are found there, right? Versus something that gives hydration to the skin, right? Dermatan sulfate. In some matrices, you might need heparin sulfate, right? Or hyaluronic acid, right? All of those are important, water holding molecules that are classified under proteoglycans. Okay. They're hydration structures, they're highly negative, right? And they're made of GAGs, right? Glycoproteins, on the other hand, they're very different. Okay. Glycoproteins are usually more on the protein base, right? Whichever is the suffix, that's where the most of the structures are. Okay. So glycoproteins means proteins are more, glyco part is less, right? Proteoglycans means glycans are more, proteo part is less. Okay. What are these made of? Just short branched oligosaccharides. So few carbohydrates getting attached to amino acids, and then you call them glycoproteins, right? We'll see a couple of formation, the Hunter-Hurler syndrome and all of that. That is where glycoproteins and things may refer to. Okay, so they may help in signaling, recognition, and all of that. Okay, now going forward, we're not gonna sort of go into the depths of it, but I've kept it here to help you create associations. Okay, so just so even if you have not looked at glycolysis in a while, you see glucose going into this pathway right here, eventually making fructose 6-phosphate, right? In this case, what we've done with fructose 6-phosphate is we've attached, you see, a glutamine and given that amine group to that fructose 6-phosphate, right? So aminotransferase, I know it's not clear, but aminotransferase means NH3 has been transferred, right? You make a glucosamine, right? Eventually, all of this nuisance will make glycosaminoglycans, right? GAGs, okay? It will go into making GAGs, right? So what we've done is we've combined a carb, which is fructose in this case, and we made like amino acid sugar. Does that make sense? Amino acid sugar. So that's what we made here, right? UDP glucosamine. Couple of other types of amino acid sugars that you may see. You see, you go down this line and you make something called N-acetyl neuraminic acid, nano. Okay, you make nano, and eventually you go into this pathway, which is the sialic acid gangliosides. You see a lot of those lysosomal storage diseases. Why are we learning these guys? Lysosomal storage diseases are where you see this. Okay. Lastly, making UDP N-acetyl glucose galactosamine, NAG. Okay, NAG. So you made NANA, you made NAG, right? And all of these can make glucosaminoglycans and glycoproteins, both, right? So this is the core sort of chemical pathway making all these structures. And if you're not able to destroy them, then you get the lysosomal storage disorders, right? So I'm giving you context, and eventually we'll build on them one by one, right? Take a minute, absorb this, and then move forward. Okay. Now, as you see here, this is the bond we made, right? This is the sugar, right? And then this is a N-acetylated sugar, which means this is the part which is combined to that sugar, the amino acid that is combined to that sugar, right? I have just made an amino acid sugar, right? So that's the basic bonding. And then you see N-acetylated, it can be O-acetylated. Like in this case, the amine group is the thing that made the bond, right? What if it was the carboxyl group that made the bond, right? Then in that case, it wouldn't be O-acetylated, right? Not to be, too nuanced with it, but these are things that they might sort of end up asking in some cases. Okay. Now you see, this is an amino acid sugar. You see, NH2 is attached to a glucose molecule, glucosamine, right? Glucuronic acid. You see glucose molecule, but with the COH. It's not. It should not be there. It should only be like this, CH2OH, right? Now you have a COH, glucuronic acid. Then they may say L-eduronic acid. You see, it's the same thing, but now the positions are different of that COH, right? Let me just show you this because you may think I'm, teaching you irrelevant stuff, but just to show you lysosomal storage diseases. Right? Here is, right, all of these, right? The L-iduronidase, right? Iduronate 2 sulfase, right? What are they talking about here? This is what they're talking about. Okay? This is the molecule that they're talking about. They're not able to break that down, right? And we'll see, right? We'll see all this. But remember, give biochemistry is best learned when you give them pictorial representations. Give your mind something. Otherwise, it just sounds like a bunch of gibberish coming together, right? Give your mind pictorial diagrams to help you remember these things. Okay? So glucosamine, glucuronic acid, and then iduronic acid. Okay? Now, let's get into it one by one, right? You see what's happening in this case. All the different like the proteoglycans are mentioned here. Okay. And these are actually glucosaminoglycans, right? And then GAGs will make a proteoglycan. So we'll look at the structure. But imagine these are all the different amino acid sugars coming together, forming special types of bonds, right? And having special type of characteristics. Okay. Do we have to remember the bonds or do we have to remember the sulfur positions and all of that? No. Okay. That's not required, right? But what is required is it's if they, end up saying dermatan sulfate or like heparin sulfate or whatever, right? You should be able to recognize that that is a glucosaminoglycans. It can be found with proteoglycans, and then proteoglycans have special functions. Okay. So just quickly going over the functions of these, you see chondroitin sulfate, right? They make up cartilage. They bind collagen. Collagen is a protein all on its own, which we'll look at, right? But they bind the collagen, hold fibers in a tight, strong network. You see, the matrix is one thing, right? These guys are the matrix, right? They make the matrix. And the collagen fibers are, binding to those matrices, right? So you see found in cartilage, found in bones, ligaments, aorta, right? If there's a problem in this, you may not have strong cartilage, right? And that could lead to some sort of pathology and questions all on its own. Okay. Moving forward, keratin sulfate, right? Keratin sulfate, you see the structure here. You have 2 sulfur molecules, different type of beta bonding, and then different type of amino acid sugar, right? Do we need to remember this for the exam? No. We do need to remember the functions at least, right? Keratin sulfate, used in usually like, keratin-like structures, right? So keratin sulfate 1 used in corneas, found in corneas, right? Keratin sulfate 2, loose connective tissue, usually maybe areolar tissue or other type of tissue. Okay, that's keratin sulfate, right? Hyaluronate, a lot of skin products and all this, they promote like hyaluronic acid, right? What is it actually made of? You see, glucuronic acid, right, with the CO minus, and then N-acetyl glucose, right? NAG protein, NAG amino acid, sugar, right? And then they make that bond and they make hyaluronate, right? Eventually, as a lubricant, as a shock absorber, synovial fluid is made of it, vitreous humor is made of it, umbilical cord has it, loose connective tissue everywhere has it, cartilages have it, right? Hyaluronic acid. Okay. Going forward, dermatan sulfate sounds exactly what it does. Like, it's in the skin, it's in the blood vessels, it's in the heart valves, right? As you go down, heparin. You see, heparin structure is kind of given to you, right? Serves as the anticoagulant, and we know most of these. Okay, and then heparin sulfate. It's you may confuse it with heparin, but this is seen with extracellular material. Okay, heparin sulfate is a extracellular material, may be found in basement membrane. Okay, not to confuse the two. Right now, we synthesized it. Now, what we're gonna do is the problem that actually comes is you can have problems making it, right? That's a deficiency, but you have problems destroying it. That's an accumulation, that's a toxicity. Okay, I've given you four, right? Hunter, Hurler, Sanfilippo, and Sly, right? The thing is, if they really want to make a tough exam, they'll give you all four in that one option, and then you're like, this sounds like Hunter, let me go with that, right? So understand all four of these and understand how to differentiate them. Okay, now, as I said, right, we looked at iduronic acid up there, right? Iduronic acid has a sulfur group in it. If the enzyme iduronate sulfatase is not able to take out the sulfate out of that, then you get Hunter syndrome. Okay, that's how easy it is, because now you know the structure, now you know the bonding, now you know the sulfur in it. Now it makes sense. Okay, what is the problem that you see here? No corneal clouding, but physical deformities and developmental disability. Right, milestones will not be achieved. Right, physical attributes are very typical physical attributes that we're going to see here. Okay, so you can't degrade your dermatan sulfate and your heparin sulfate. So you can understand it. Cells need an organized basement membrane, right? You have just affected the basement membrane problem, right? Basement membrane has a problem now. Okay, that is Hunter. Okay, what is Hurler? Within that same sort of sequence, if I'm not able to break this iduronate down, then I get Hurler. So one was taking the sulfate out, the other one is just taking the amino acid sugar out. Okay, where you see here, coarse corneal clouding, right? Coarse facial features, and then of course, a couple more physical deformities. Okay, dermatan sulfate and heparin sulfate both are affected because the sequence is seen in that most common. Deposition in coronary artery leads to ischemia and early death. You see, again, MI, right? If someone is showing with heart problems and MI problems early on, right, and then they talk about coarse facial features and stuff, you have your diagnosis. Okay. Lastly, let's just look at Sanfilippo and Sly so you can rule these two things out. There's nothing new to you. Okay. Am I asking you to remember it? No. I'm asking you to differentiate from Hunter and Hurler. Okay. So Sanfilippo, you see all of these different types are there. Okay. A lot of them are focused on nervous system disorders and developmental disabilities. Okay. Do I have to remember all of this? No. Right? You see heparin sulfatase not taking out the sulfate, not taking out some coenzyme, or not acetylating, etc., right? Do we have to know all of this? No. Nervous system based, right? Do these two have specific nerve problems? I don't think so, right? They have developmental delays, right? But nervous system disorders, maybe not. Okay. Sly disease, right? Glucuronidase, right? Beta glucuronidase, taking out that glucuronic acid, right? You see, in this case, hepatosplenomegaly, skeletal deformity, short stature, and corneal clouding. So Sly kind of starts to look like Hunter and Hurler together, right? And you see, again, degradation of dermatan sulfate and heparin sulfate. So all three of them can start to look alike. But this looks like both of these guys, right? With the hepatosplenomegaly. These guys won't have the hepatosplenomegaly. Okay? So just put some pictures to the things that you've learned. You see, this is Hunter syndrome, right? You see the physical deformities and all the list of it, right? And then go back and revise it again from first aid. Okay. Now you look at Hurler syndrome, right? You may see hepatosplenomegaly here. Facial features, eyes clouded, short stubby fingers. That's just a mnemonic for you. But I would still say, keep to this diagram and the first aid. Okay, I've given you this so you can, imagine what it looks like. Okay, so that was glucosaminoglycans. Glucosaminoglycans, they congregate together and they make this big bottle cleaner like protein. This is a proteoglycan. This was the thing that we're making all this time. Okay. You see, this was chondroitin sulfate. You see how it's making the chains, and eventually it's connected to a core protein, which is connected to a backbone. Usually it may be hyaluronic acid by itself. Okay. So all of those are coming together and making this, right? And I've given you the bonding. For example, if this was the core protein, then you have all these different linkages going from inside to out, right? This is what they're talking about. Serine, protein, side chains, linkages of xylose, galactose, and then eventually the amino acid sugar that we talked about, right? So basically, repeated chains of these, right? That's what we made here. That is a proteoglycan. All right, I hope you understand it. Now we can sort of go forward from the whole mystery of Hunter and Hurler and what is going on, right? Next is, as I said, end-linked glycosylated protein. So this is actually glycoproteins, right? You see that long protein structure and then a small, oligosaccharide structure, right? Cell signaling, very, very important, right? You see side chain of asparagine, which is, an amino acid, and then you see all that binding, okay, the glycans. All right, so this is just a schematic diagram of how these different glycoproteins are made. Keep it simple. Ribosomes make the protein, and then rough might glycosylate them, right? It's making all the oligosaccharide structure, and then just attaches it. Then eventually sends it to the Golgi apparatus, where you can, secrete it or package it and send it wherever the cell needs. Okay, keep it simple. Don't confuse all of this, right? Peptide grows, attaches the carbohydrates, sends it to Golgi, and now Golgi can package it and distribute it everywhere. Okay, Golgi is FedEx. Okay, now because there's different coordination required in this whole structure, there's a thing called eye cell disease, right? It is mentioned in the first stage, so I'm going to go to that page. You see, rough transmitting all these different vesicles to the Golgi, and the Golgi packaging and them to the outside with the plasma membrane. Okay, if plasma membrane gives a vesicle, if Golgi receives it and it needs to send it to the rough it can do that, right? There's two types of proteins that help in signaling and communication between Golgi and RER, right? It's given right here, COP1 and COP2, right? Do take a look at it. These are the trafficking proteins, right? Now, what we're talking about with eye cell disease, right? Inclusion cell disease, okay? It is a lysosomal storage disorder again, right? But in this case, what's happening is this molecule right here that you see, NAG phosphotransferase, right? This does not allow mannose residues, right? What you're supposed to do is you're supposed to make mannose 6-phosphate, right? You're supposed to phosphorylate your mannose. Mannose is, like a carbohydrate, right? You're supposed to phosphorylate them on these glycoproteins, okay? Now, you see, as this diagram shows you here, you made this thing and you have a lot of mannose, right? Typically, what's supposed to happen is you're supposed to phosphorylate your mannose in order to activate and send it out, right? So they can function, right? And within the eye cell disease, you're not able to do that. And you see this mannose 6 receptor that was supposed to detect that's not receiving its signal. So what ends up happening is this, right? It attaches to a lysosome, right? And it's never sort of destroyed, right? It's never sent out, right? So the transport vesicle is just sitting with the lysosome, and there's no activation of any acidic or not even transmission of it outside the cell. Okay, so you just created something that is not going to be used, and it's going to be sitting in your warehouse. Okay, I hope that makes sense, right? So within the cells, you see if you start to accumulate these glycoproteins over and over and over, right? So you see buildup of cellular debris in lysosomes. These are called inclusion bodies. Results in all these different physical deformities. Okay, gingival hyperplasia, important. It's not just phenytoin or calcium channel blockers. You have a disease now, right? Corneal clouding, similar to Hunter. I think it was Hurler on my bad, right? Restricted joint, so actual bony deformities, right? Kyphoscoliosis. Okay. Increased plasma levels of lysosomal enzymes. Yes, they can spill over into your blood. You may detect them, you may not, right? Symptoms very similar to Hurler, right? But it's more severe than Hurler. Okay, so that is eye cell disease. I hope that makes sense. You're not phosphorylating your mannose, and eventually that trigger that's supposed to let the mannose-6 receptor and let the Golgi know to, transport it, right? It's not happening. It tries to mitigate it by sending it to lysosomes, and the lysosomes are not degrading it. Okay, so I've given you the diagram here too, right? So a lot of lysosomal storage diseases we've covered. All right, moving forward. Now, another special type of protein, okay? It's important to know these proteins, okay? I've given you a decent amount of proteins in this lecture, okay? First one being heme, right? You start heme by aminolevulinic acid, right? You take two molecules of it, you dehydrate them, and you make this first thing, which is porphobilinogen, okay? Now, how does this ALA is actually made? This actually comes from succinyl-CoA and glycine. Glycine is another amino acid, and even this I think started as an amino acid. And you attach a coenzyme A to it. Okay, so these two amino acids made the heme, the primitive heme structure. Okay, ALA makes this porphobilinogen, and then eventually you make all these different patterns, and you end up making hemoglobin, right? Where the iron is going to sit inside this, forming that 3D structure, right? And where the iron can actually bind to, oxygen. Okay, so I've given you the main things. Just to review it, you see, here's all the enzyme names, right? ALA synthase, that's the first one. Dehydratase, if you can remember it. Porphobilinogen deaminase. Deaminase just means taking out the amine group, right? You come here, you get UROD, uroporphyrin decarboxylase. Then lastly, if I can go down, I think so. Pyroketolase. Okay, so the ones that are bold, that you have to remember. Okay, and then the related porphyria is also given, right? X-linked, AIP, porphyria cutanea tarda, and protoporphyria. Okay, now let me give you a general rule to help you understand this thing. Okay, before this reaction, okay, porphobilinogen deaminase, right, which is linked to, where is this? Yeah, which is linked to AIP, right? Up until this whole structure that you see here, right? This is the thing that goes and deposits in underneath the skin, right? This deposition doesn't actually react with the UV light, okay? But this structure right here and every subsequent structure after this, right, goes and reacts with the UV light, gives you skin problems. I hope that makes sense, right? So everything after AIP, right? Everything after this PBD, I call it, right? Porphobilinogen deaminase, right? Everything after will give you skin conditions. Everything before, no. Okay? Understand that clinical point. It'll help you remember things better. Okay? I've given you a diagram right here. Let's go through it one by one. Let's start with, and you see, I've not mentioned it before, but the formation of heme happens between mitochondria and cytosol, right? It starts with the mitochondria, goes intermediately into the cytosol, and eventually comes back into the mitochondria itself. Okay? So you see, you start with this thing right here, right? Succinyl coenzyme A and glycine. You make ALA. ALA goes to the outside, makes PBG, right? Eventually, what you're gonna suffer with if you have a problem here, you have AIP, right? Now, because you're making all of these things, patients are not photosensitive. That's the main thing you should remember. Okay, you're gonna get a lot of different signs and symptoms. Skin is not gonna be one of them, right? And I'll go to that page in first aid to elaborate more. But AIP, that's why that's special. Okay. They've also given you lead poisoning. Lead has a couple of different enzymes that it inhibits. One of them being ALA dehydratase, right? So that very first step. Okay. Last one is pyroketolase, which is the last step. So first and last is lead poisoning. Okay. Now, of course, what ends up happening is if I'm, you know, inhibiting it here, right? PBD is inhibited, then this will spill into the urine. Okay. Now, the thing is, this thing oxidizes, right? If I show you the structure again, you see this thing oxidizes. Nope. This thing can oxidize, right? When it's exposed to air. So it may show you, you see, urine darkens on exposure to light and air. That's a special thing that AIP people will show, right? Because the structure is not stable, it can oxidize and it can turn into sort of a dark color, right? Another important point. Now we come into the Porphyria cutanea tarda. I don't know why it's doing this. We'll just use this. All right, so now we made hydroxymethylbilane and then eventually uroporphyrin, right? And then eventually you're supposed to make this whole cycle right here. Okay. Let's do PCT first. So Porphyria cutanea tarda. Chronic disease can be autosomal dominant. Uroporphyrin accumulates. It is the most common type of porphyria, PCT, right? It's in the name, cutaneous problems are going to show, right? Patients are photosensitive, okay? Everything is stopped here, so everything that is in the back, right? They will all be shunted, right? So this is uroporphyrin. This is basically like like urine color, or corpophyrin is like stool coloring, right? So they're shunted into these directions, right? So that's what's gonna happen. But are you gonna make heme? No, you're not, right? You can look at congenital erythropoietic porphyria. Again, photosensitive, right? Accumulation in the urine, right? And then I'm pretty sure they have like some sort of bone marrow issue. Okay. Finally, they've given you a couple more variegate porphyria versus all of the. You see, all of these are photosensitive. Patient photosensitive, right? Whereas the other ones, they're not. All right. I hope that helps. Let me just go to the first stage part of it. Here we are. Just looking at it briefly, right? Lead poisoning, AIP, these are all the system is affected, right? It accumulates and causes that problem. Port wine stain, port wine urine. That's what they like to call that, dark urine, right? But porphyria cutanea tarda, now it's sitting in the skin and causing skin problems, right? Do go over this. This will help you. All right, good. All right, now just to reiterate on like the bile pigments and how they're sort of formed. We made hemoglobin. We should understand how to excrete it, how to destroy it and excrete it too. Okay. The couple of enzymes that are responsible and they're very important. First is heme oxygenase. Okay. This thing is directly linked to heme, right? Because how else are you going to get, how else are you going to explain intrinsic versus extrinsic hemolytic anemia, right? Or extravascular versus intravascular hemolytic anemia, right? And this enzyme is very, very important for that, right? All of the extravascular hemolytic anemia, right? Anything that is destroyed in the spleen or the liver, right? That is extravascular, right? Those are going to undergo, the macrophages are going to eat the RBCs, and they're going to use their heme oxygenase. Eventually, you open up this flower chain into, you just open it up from one side, cleave it and open it up, and you get this long chain like this, right? Eventually, you will reduce it using energy, right? You will reduce it, biliverdin reductase, and you make bilirubin. This used to be green, this used to be red, right? You take that bilirubin, give it to the liver, it makes the conjugation happen and secrete it into the bile, and then fatty acid metabolism. Okay. One clinical correlate that they always like to ask is a newborn, right? Especially someone who's not full term, right? How are they dealing with the bilirubin and bilirubin formation? Okay, So you see, a preterm birth, you may not get enough of UGT activity. That means the conjugation activity. It's always going to be low, right? Even with this, right? Serum levels of bilirubin rise in a premature patient higher than a full-term patient, right? Because the UGT is not working too much, right? That's why they give them light therapy. Okay, so that was about the story of heme. Okay, now comes the story of collagen synthesis. Very important, right? Very important. You see, collagen is made up of, again, amino acids, and the two of the most important sort of, amino acids that they will love to ask you about is proline and lysine, right? Proline and lysine are supposed to undergo hydroxylation, right? And they're supposed to make that cross links within the collagen, right? Here is the collagen formation. I do need you to study this on your own, right? It shouldn't be too difficult, right? It shouldn't be too difficult. Now that you've understood, and I will also use page 69, right? All of this should not be too difficult. Have to pay attention to the cofactors though, right? And you see hydroxylation of proline and lysine under the jurisdiction of vitamin C. Glycosylation, adding it with the sugar, and then pro-collagen, and eventually the final sort of maturation of collagen happens outside of the cell, right? Because how can you get a bunch of collagen, and cross-link inside? It can't, right? You have to get it outside. So you have to cut the ends off, and then when you got all that bunch of collagen, then you can cross-link it, right? And then it's stabilized by lysyl oxidase. That's the enzyme. Okay. This is related to copper, and copper is related to Menkes disease. Okay. Menkes disease, a lot of this I'm going to go over when we do like vitamins and minerals lecture. So I'm not touching on it right now. But collagen formation and, the relation of collagen with like Marfans and other like osteogenesis imperfecta types of collagen, all of that, right? Please go over this right now. Okay. All right. Next comes insulin formation. Okay. Now, again, very, very important peptide chain. What you see here is proinsulin, and then insulin. Three things. Do not get confused in either of them, right? Pre-pro does not have any internal bonding with itself, right? This is just plain chain, okay? Once you start making the disulfide bridges between the two, and where are you making the disulfide bridges? Cysteine, right? You make cysteine disulfide bridges, and eventually you end up with the secondary structure, which is proinsulin, right? Finally, what you're going to do to make insulin is make a cut by this region, right? And then you get a C-shaped peptide, C-peptide. And then you excrete it. So this is the diagram you see, starting from the nucleus, coding, making the amino acid, making the proinsulin, and eventually you're making it and excreting it with the C-peptide and the insulin in the bloodstream. Okay. Now, I hope that if you understand the process, then you'll understand exogenous and endogenous production of insulin, and they'll have to ask, which one's which. Okay. I hope everything makes sense so far. All right, guys. The next topic we're gonna look into is transamination reactions. Right. So we've seen, or you must have heard about ALT and AST and all of that. Now we're just gonna study the biochemistry behind it, because they do like to ask these things and correlate it with parts of like glutamate metabolism, because that's the main thing. Goes into urea cycle, the disorders of urea cycle, and keeps on going with other amino acid pathways, right? So let's look into transamination. The first thing is you need an amino acid, and you need one type of alpha keto acid, right? Now, transamination takes one of the amine groups, gives it to the other group, ends up making a new alpha keto acid from one backbone, and ends up making a new amino acid from a different backbone, right? So you see what happened here is alanine ended up giving this ammonium to alpha ketoglutarate, making glutamate, right? Whereas the alanine itself high, ended up getting a ketone group attached to it and became a pyruvate. Okay, that is ALT. That's the function of ALT, right? Transamination means you take a amine group from one um, substrate into another one. Okay, now ALT is just that glutamate pyruvate exchange. Okay, now when it comes to AST, and this is mislabeled, but AST is oxaloacetate instead of that alpha ketoglutarate, right? So it uses glutamate and oxaloacetate. Okay. Now let's just differentiate them quickly so we understand exactly what's going on. ALT, you see, ALT is more liver specific than AST, right? If you have ALT being elevated, you are sure to know that hepatocytes are being damaged, right? And the reaction is between alanine and alpha ketoglutarate, and the AST is between aspartate and alpha ketoglutarate, right? AS aspartate, okay? ALT, okay? Ends up making a pyruvate and a glutamate. Here you make oxaloacetate and a glutamate, okay? And you can read the clinical significance. You see AST is kind of non-specific compared to ALT, right? But both of them can be increased if you have hepatocellular damage, right? Now, why did we end up doing this whole transamination business, right? The thing is, amino acids and peptides and enzymes, they grow old. They need to be broken down, right? Now, what is the main carrier protein, main carrier group that takes all this amines, right, from the amino acids and neutralizes them or recycles them, right? That is going to be your glutamate. That's the name of the game, right? Glutamate is going to accept all these different amine groups from different things. For example, alanine giving it NH3, right? Plus, or aspartate bringing it NH3 plus, right? Because once you have glutamate, glutamate can then go into the liver and it can be, it can undergo this process of oxidative deamination, right? Which just basically means that you take the glutamate, you use, you end up oxidizing it, and you end up sending it into the urea cycle, right? So what we did was we took amine groups from different amino acids in order to recycle them. The main character in this scenario is glutamate, and glutamate is eventually dumping that ammonium into the urea cycle, right? That's what we're doing. So this is also called the glucose alanine cycle, right? If you've seen this diagram in the first aid, this should ring some bells. So as you see muscles are constantly in synthesis or destruction mode, right? Especially destruction if you're performing gluconeogenesis. They take all this amino acids and they turn and make glutamate, and they end up making or end up sending alanine to the liver, right? And then alanine, once in the liver, makes glutamate and sends it to the urea cycle, right? And then you excrete it out through urine or whatever. Okay. A secondary cycle is called Cori cycle. Cori cycle is completely different, but again, this is used for energy, and this is used for usually excretion, I would say. Okay. So Cori cycle is just simple. Let's say you're working out anaerobic workout, you're building up a lot of lactate. Lactate can go back to the liver and it can be turned into glucose. That's it. It's simple, right? So when you're running for, let's say you're running, you start running for like 5 minutes or something, right? Within the second minute, let's say you got, tired, okay? But then you stop for maybe 30 seconds and you're back to normal, back to good performance level. The reason for that is the Cori cycle, because your body started making lactate. You stop for 30 seconds, all that lactate went into the liver, and the liver make glucose, and they send it back to the muscles. So you see the second wind phenomena, or like just overall energy monitoring under anaerobic conditions, is Cori cycle. Cahill cycle is the transamination excretion of the main function is the excretion of the ammonium. Okay. So now let's just consolidate all that we've learned into these diagrams here. Okay. So as you see, the cell, sorry about this. Let's zoom in here. Right. So cell is coming up with these ammonium molecules. Glutamate transfers them. Now the thing is, glutamate can turn into glutamine, right? Glutamic acid, glutamate, right? You add another ammonium on it, you make it glutamine, right? Glutamine can go to the blood and go into the liver, and then you can recycle it and make the glutamate again, or and send that ammonium to the urea cycle. That's one way that glutamate does this business, right? Attaching itself to the amine group and then transporting directly. Okay, let's see what the next way is. We know this next way already. Take it and combine it with some sort of alpha ketoglutarate. It can be ALT, it can be AST. Go through the blood, go into the liver, and eventually make glutamate and and send this ammonium somewhere else, right? So that is ALT. Now, as you see in this diagram, once the glutamate is here, right, it can undergo into two options, right? Two cycles. It can go and make ammonia go into the urea cycle, okay, or it can go and make aspartate. And the aspartate itself also goes into the urea cycle, and we'll see where that happens, right? So all that we've learned so far is how does a cell or how does a muscle send its degraded products, right, which is mostly ammonium, into the urea cycle. This is how they do it. Okay, so that is the main role of glutamate in all of this. Okay, now we're gonna go into the urea cycle, but I just wanted to point out one enzyme that you need to sort of correlate the next urea cycle with. Okay, the main thing is CPS2, carbomoyl phosphate synthetase 2. Okay, what is it taking? Glutamine and carbon dioxide and making carbomoyl phosphate, right? This is the brother of CPS1 that you see in the urea cycle. Okay, so carbomoyl phosphate synthetase 1, right? You see here it took CO2 and it took the ammonium, right? Usually it's glutamine, glutamate, whatever, right? And eventually it goes in and makes carbomoyl phosphate in the liver. So do we understand this, right? So there's CPS2 here, and then there's CPS1 here. Both of them are performing the same thing, right? If there is, let's say, a problem with CPS2, what's going to end up happening? Will you be making your pyrimidines and purines? Probably not, right? But then all of this will be shunted to the urea cycle, and vice versa. Let's say there's a problem in the urea cycle. What do you think is going to happen? What do you think this first enzyme is going to do? It's going to make the carbomoyl phosphate, and that carbomoyl phosphate will go into making purines and pyrimidines. So both of these brothers are playing the same role, right? And they end up switching if one of them is going haywire, right? So we need to understand that. And as I said, looking out for aspartate, you see glutamine here, which is a derivative of glutamate, and then you see aspartate again, right? Both of them are like acidic acids that we talked about. You come back here, right? You see CPS1 makes carbomoyl phosphate, does all of this. You see aspartate enters the chat and has caused arginosuccinate formation. Okay, so carbomoyl phosphate, glutamate, aspartate. Okay, now let's take this one by one. Here's the familiar diagram if you've seen it in first aid. Okay, first thing is make citrulline, send it to the cytoplasm, make all the other things like arginosuccinate, arginine, etc., right? While taking in the aspartate, and eventually you make the urea, which is this structure, NH2CO NH2, and you send it to the kidneys for excretion, right? Or regulation. So that is the urea cycle. Okay, so just looking at try to come up with the structures. It will help you remember it better. You see citrulline, right, with the NH2 group. You take the aspartate from the other one, and now you combine them and make this compound, which has both the nitrogen compounds. You excrete out fumarate in the next reaction, and you only have this whole arginine thing right here. Okay, you made arginine. Eventually, you're gonna cut this part out, and you combine it with water, and you make NH2CO NH2. Okay, that's urea. Now we understand this. Let's look at some of these deficiencies and the things they like to ask you about. Okay, first thing is called OTC deficiency, and this is one of the major ones. You may not see the other types of urea cycle disorders, even though there are many. This is the main one that they want to talk about. Okay, so Ornithine transcarbamylase amylase deficiency. You see, this is the thing. You're not making citrulline, right? You're gonna make carbamyl phosphate. And what did I say? If CPS, if this thing is working and you're accumulating carbamyl phosphate and it's not going forward, what will this do? This will go into pyrimidine and purine formation. You need to understand that, right? Once you send this into purine and pyrimidine formation, what are you going to end up doing, right? You will start making the reaction that you see here, right here, orotic acid. You see, everything is, you have a lot of this forward flow. You make orotic acid. Okay, so what are these patients going to present as? You see, high orotic acid in the blood. But can you make urea? No, so that's low. And what is happening to the ammonium? That's bleeding. It's going into the plasma freely. There's nothing else that you can do. You can't excrete it out, so it's building in your blood. I hope that makes sense, right? Everything else is just explaining that part of it, right? And if you understand the diagrams, if you look at the diagrams once over again, you'll understand it completely. Okay. Now, similar to this, there's something called orotic aciduria that you see here. That is also simple. What's happening there is that this enzyme right here, UMP synthase, right here, UMP synthase is impaired in orotic aciduria. Well, that's just easy. Orotic acid will be increased, but will there be a problem with urea formation? No, everything is working here fine, right? Will there be a problem with hyperammonemia, something like that? No, because urea is being made. Everything is going to be okay. But because of this orotic acid, right, forming, is anything else forming here? No, you're not making any DNA. And if you don't make DNA, can you mature your RBCs? No, they're nice and big. It's causing megaloblastic anemia. Simple, right? Similar to vitamin B12, similar to vitamin B9, it's causing megaloblastic anemia. So if someone comes to you with megaloblastic anemia, right? They have no signs of vitamin B12 deficiency, no signs of folate deficiencies. They're not on any drugs or they don't have any autoimmune condition that can cause vitamin B12 deficiency. Think about this. Okay. So that is the difference between orotic aciduria and OTC deficiency. I hope that makes sense. Right? Let's read one of these cases. 7-day-old boy is brought in for evaluation of poor feeding, lethargy, vomiting, and irritability. Right? Physical examination is significant for hypotonia and hepatomegaly. They do lab testing. It shows high ammonia, low citrulline, and high orotic acid. That is easy, right? That's an easy thing. If they ever said just low citrulline and maybe orotic acid, you might have to, think about it. But this is easy. Okay, what are you going to do for them? Limit the protein intake, advise high calorie diet, and amino acid supplements, right? Because they might not be using and making the right amino acids, right? So control their intake and add these supplements. All right, now we're going to talk about one high yield thing. Okay, it's important to distinguish between these three things that you see here. Okay, ammonia, uric acid, and urea, right? Hyperammonemia, hyperuricemia, and uremia. That's all this is. Uremia. Okay? These three conditions, students blunder them all the time, right? And you need to understand your way out of this. You should be able to, right? Now, this is the section in the first aid, right? And you can go over this. Main thing I want you to focus is on this part of the diagram right here. Alpha Kg, right? If acquires ammonia, goes into glutamate formation, typical transamination behavior. But if glutamate acquires ammonia, means becomes glutamine, or glutamate can go and make GABA, which is a inhibitory neuron. Right? Now we're gonna look at the treatments and all of that, but first we're gonna understand the fundamental differences in all of these. Okay. Now let's look at the presentations for ammonia, hyperuricemia, and uremia. Okay. Let's go from one system to the other, right? CNS manifestations. Hyperammonemia people will show up with cerebral edema because their blood-brain barrier is being damaged, right? Ammonia is a simple molecule. It can cross the blood-brain barrier easily. Okay? And if it does that, you have a problem. You have something called hepatic encephalopathy, right? The usual asterisks, confusion, seizure, all of that is ammonia. Uric acid does not lead to any neuro symptoms, right? Usually in the peripheral and usually makes a crystal, right? As gout. Uremia, on the other hand, can lead to confusion, asterisks, seizure, and coma. You understand? That's the main thing. Uremia can cause problems, neural problems, right? And they've given you a couple of like mechanisms: astrocyte swelling, and then some sort of, intermediate formation and molecule formation, and then that goes and damages neurons. But at least knowing that these two are the ones that cause neuro problems. Joint problems. Who's going to be the main thing that's causing joint problems? It's the middle one, uremia, right? Uremia causing gout and destruction of the cartilage and all that. We understand that. Uremic arthropathy, right? And where do you see urea being increased? Renal failure. So imagine this as a renal failure patient. Imagine this as a gout patient. And imagine this as a hepatic encephalopathy patient, right? So when you look at uremic arthropathy, you see beta-2 microglobulin, amyloid deposits in joints, maybe renal osteodystrophy. All of that is related to just renal, problems, renal issues. It can lead to some sort of crystal formation, but that's not, well established, right? Are they, can they lead to some sort of joint involvement? Yes, that's the only thing you should get from this. Cardiac. You see, not much, from it directly. Indirectly, though, it may lead to coagulopathy because hepatic problems, right? It may lead to hypotension, again, because of hepatic problems. Why? Because you have like, vasodilators floating around in the blood because the hepatic system is not, metabolizing them. Okay. Hyperuricemia, you see, you may have endothelial dysfunction. That is important, right? Coronary artery disease and all of that is a risk factor. Here you have a special sort of thing. You have pericarditis. That's the first thing and main thing you should remember, pericarditis. You have accelerated atherosclerosis, maybe left ventricular hypertrophy, maybe heart failure. But at least, right, with renal failure patients, we have to remember uremic pericarditis. Next, GI stuff. You see, all of them can lead to nausea. All of them can lead to, vomiting feeling. This jaundice, especially ascites, portal hypertension because hepatic dysfunction. Can this lead to, feeling of any like liver related issues, jaundice? No. Okay. You may get urea crystallization on the skin and other things in uremia, right? Renal failure patients. But apart from like nausea, vomiting, you may not see anything, right? You can have this ammonia breath on their physical examination, but apart from that, nothing, right? Take care of the lab markers. LFTs are going to be elevated, ALT, AST, blood pH, depending on what they have, right? Here, nothing major. Here, everything that is renal problem, right? Renal markers are going to be elevated. Imaging, you're going to see maybe brain and MRI, X-ray, you may see erosions in the joints. Here, renal ultrasound, shrunken kidneys, echocardiogram, that's what you're going to do. Now they've given you management, right? And we know all these, sort of overlap. You see dialysis for the renal patient, NSAIDs, colchicine extra for the hepatic patient, and lactulose for the ammonia patient. Okay, so you see, just a last sort of discussion on that topic. Chronic kidney disease can impair the urea cycle, which causes amonemia, right? So you can have both amonemia and uremia, especially in a patient who has renal problems, right? And if you end up having that advanced liver failure, you're gonna start to mimic urea because you may have hepatorenal syndrome, right? So if renal failure can lead to hepatic problems can lead to renal problems. That's what they're going for. right? Gout nephropathy can progress to uremia. So, there's certain scenarios where all three of them can be there, right? So when you look at bun creatinine next time, or when you look at, uremia, or they talk about hyperammonemia, you should be able to differentiate them in your head. Okay, so that's our discussion on that. Next, we're going to move forward and we're going to look at what amino acids can make, right? What other products can amino acids form within the body, right? Now, the main first aid page that I want you to look at is mentioned over here. Okay, first is the amino acid derivatives right here, and the next one is catechol metabolism, like right here. Okay, as you see here, the main factor that you are encountering is BH4. It's called tetrahydrobiopterin. Right, this is the thing that reduces. So if I show you this, if you see this now, right, you see phenylalanine is making tyrosine, right? What's the difference between the two? It's this OH molecule, right? Tyrosine to L-DOPA. Now it's OH molecules again. You keep going, and then you fix the dopamine, norepinephrine, etc., right? But tetrahydrobiopterin is the common cofactor, right? So they've given you all these major reactions that use tetrahydrobiopterin. Try to make intuitive sense of this, right? We've already seen tyrosine, right? Tryptophan making serotonin requires tetrahydrobiopterin. Nitric oxide for vasodilation requires BH4, right? Alkyl glycerol making fatty aldehyde. I don't really remember where this is, but BH4 is indicated. So at least these three you're gonna have to remember. Okay, that is tetrahydrobiopterin. Now, if I'm saying that this is required for reduction, it itself is undergoing oxidation, right? So who is the one thing that regenerates tetrahydrobiopterin, right? It must be a reductase, right? So the enzyme that brings or regenerates tetrahydrobiopterin is tetrahydrobiopterin dihydrobiopterin reductase. Okay, I think I mentioned it somewhere. We'll get to it. Okay, but there is a reductase enzyme that regenerates all these things back, right? So first, we're gonna look at all these different stuff that you have, I'm sure, encountered in pharm or psych and all of that, right? So let's just review it quickly. We've seen the structures. Phenylalanine, PVT Tim Hall, right? P, phenylalanine is essential. If this is essential, then its derivatives are non-essential. Tyrosine is non-essential, right? If someone has phenylketonuria, right? That means then they cannot make the convert phenylalanine to tyrosine, right? That means this becomes a essential amino acid for phenylketonurics. Okay, simple. If you cannot convert it, right, this is non-essential now, and this is essential. You have to supply them tyrosine. Okay, we're gonna look at PKU in just a moment, but we're just figuring this out. Okay, now we've seen L-DOPA formation, we've seen dopamine formation, decarboxylase. Okay, which means you're taking out a CO2. And if you look at the thing again, right, you see we're taking out this group right here. What is this? CO2 right here. And you get rid of the H, but CO2 decarboxylase, right? Then then they say hydroxylase. So what does the hydroxylase do? Add an OH group. And what's the difference between norepinephrine and epinephrine? It's the methyl group. Okay, so this N has a methyl on it, and it changes the chemical nature of the compound, right? So that's what we're going through here. So what is the main sort of main questions that they love to ask you? Cofactors. Decarboxylase, L-DOPA to dopamine. You need B6, right? Vitamin C, dopamine to norepinephrine. Norepinephrine to epinephrine, you need SAM, right? S-adenosyl methionine, right? Which is under the control of B9 and B12, which we'll see in vitamins and minerals lecture. But this is the conversion. Okay, eventually you end up making the byproducts when you metabolize them, and you see VMA and HVA in the urine. And I've given you a clinical correlate right here. I'm sure you know this, pheochromocytoma. The levels of VMA and HVA are increased in pheochromocytoma. Why? Because you have a tumor that overproduces things. Lastly, you see carbidopa mentioned here, used for Parkinson disease people, right? Inhibits the peripheral conversion of L-dopa to dopamine. Why would I do that? Because I want to give them a dopamine substitute that goes into the CNS and it's not destroyed by this peripheral dopa decarboxylase. I hope that makes sense. Okay, I need it to go to the central region. I'm going to inhibit the peripheral ones, and mostly whatever I give should go into the CNS. Okay. Now amino acid derivatives, and we're going to look at all the diseases in just a second. Okay. What are the other amino acid derivatives? You see, we've discussed phenylalanine, tryptophan. Okay. Tryptophan makes niacin, which makes the energy currency, which is NAD, and NADP. It also makes serotonin, right? Serotonin, neurohormone. Eventually, serotonin can make melatonin. And eventually, if serotonin is metabolized, it makes the indole acetic acid, right? This is the excretory product. So whenever they say serotonin, not serotonin syndrome, but like a tumor that makes a lot of serotonin, like carcinoid, right? They're going to ask for the excretory product in the urine. That's HIAA. And you see a common theme amongst all of this. If it's not BH4, it's B6. So when we do vitamin B6 and vitamins and minerals, all of these should be correlated there. A lot of neuronal function depends on B6. A lot of energy metabolism depends on B6. So a lot of symptoms of B6 deficiency will mimic that effect. I hope that makes sense. Okay, we saw how we make heme. We saw how we treat glutamate, right? Arginine we saw too. It can go into creatine, it can make urea, it can make nitric oxide, all under BH4. Right? So as we go further, you see all of these are sort of discussed. Serotonin controlling the mood, behavior, cognition, talking about MAO inhibitors and all of that. That's a farm thing. But I've given you the diagrams and the first aid sort of diagrams you should go over. Okay, right here. Now, there's this special deficiency that I've come across while doing research. It's called AADC deficiency. Okay, AADC just means dopa decarboxylase, right? So if dopa decarboxylase is low, dopa is high, dopamine is low, right? That's the conversion we're talking about. Just to go back to it, this one, right? AAAD deficiencies lead to L-dopa increasing, dopamine decreasing, right? So what are the effects, right? So you see presentation. Basically, you have no dopamine, right? Which is also the effect of what? Antipsychotics, right? So if you have antipsychotic overdose, or like a person is being treated with antipsychotics, what are the different findings that you'll see, right? So that is how you correlate this. This is oculogyric crisis, which happens after a decent amount of time has passed with antipsychotic treatment, right? And the pathophysiology is just basically based on your direct and indirect pathways not working properly. Okay, so oculogyric crisis, hypotonia, they're not able to move, right? And again, low dopamine mimics what? Parkinson type of disease, right? Autonomic dysfunction, again, Parkinson-like disease, right? Diagnosis, serum enzyme activity, dopa products are going to be there. Lumbar puncture will give you all these different neurotransmitters being low, right? So if they ever put this in the options, right, you should be prepared with how the physiology works, right? Next comes glutamate metabolism, right? We've seen much of this. You just have to review it. That's all. Similar with arginine, mostly going into these three things. They've given you what creatinine is made up of, right? arginine plus glycine making this, plus ornithine making the final product, and getting methylated. Okay. Okay. Autonomic drugs, you've seen most of these, right? I'm not going to go through them here, but I need you to correlate them because it's very important that you understand how they're formed versus what drugs can inhibit, their reuptake or formation, etc., or release. Okay. Dopaminergic, serotonergic. Same, the same process is going to happen in both of these. I'm not going to go through much because we have to talk about like dopamine receptors, serotonin receptors. There's a tons to, go into it, but at least look over the formation and the general drugs. Okay. Now we're going to finish up with the diseases section of it, right? So let's look at this case. I want you to pause it, read it, and understand it. Okay? Pause it now. All right. I hope you're done reading it. So let's just understand what's going on here. You see, 2-year-old boy, growth retardation over the last year, born after an uneventful pregnancy. Now, it's taken his parents state that it's taking much longer time to hold his head up, sit, walk, and talk compared to his older siblings. So, developmental delay. Musty body odors noticed, and then his height and weight are very low percentile, right? He also has light colored hair, eyes, and even eczema, right? So they do some testing and they eventually figure out that phenylalanine is increased, right? And they also see tyrosine and even BH4 levels are low, right? They restrict the phenylalanine, increase tyrosine, and then BH4 as cofactor, right? So what are the couple of ways that you can get phenylalanine to turn into phenylketonuria, right? The main thing is lower activity of the hydroxylase enzyme or the deficiency generally, right? It's not just the enzyme functioning. It could be the cofactor deficiency, right? Or it could be this enzyme right here, dihydro biopterin, right? Reductase, the thing that regenerates BH4. Maybe that's a problem, right? So there's a couple of different mechanisms that are going into PKU formation, right? Last one is, let's say you have high phenylalanine levels in a pregnant patient, right? If the mother has PKU, these are the congenital effects that you'll see, right? Or if the fetus itself has an inborn error of metabolism, then they will present with their own, maybe not congenital disease, but disease later on. Okay. So just to revise, a couple of mechanisms, right? PAH not working, BH4 not working, DHPR not working, or the mother having the PKU problem. Okay. What are the findings? You see, all of this is related to, major neurotransmitter not being formed, right? Skin is hypopigmented because you're not making probably serotonin, dopamine, all of that, right? No melatonin. The musty body odor is because everything is being shunted to other parts of the metabolism, etc. Right? So you see, tyrosine is not able to do any of this. One thing that tyrosine makes is T3 and T4. Right? Tyrosine undergoes iodination to make T3, T4. Right? So if that is lost, you're not going to have any of these major hormones, and obviously developmental delay. So what ends up happening is you take this phenylalanine and it just sits there, and eventually it ends up making ketones. What are the names of those ketones? You see, phenylacetate, phenylactate, phenylpyruvate. Right? Phenyl group is just a benzene ring with a methyl group on it. Right? But what are they going to lead to? High anion gap metabolic acidosis. Right? Anion gap metabolic acidosis. And just so we know, anion gap metabolic acidosis is You take all the electropositive ions minus the electronegative ions. So sodium minus, bicarbonate plus, chloride. Okay, so anion get metabolic acidosis coming up in a child, and then you see all these other developmental problems, you have your diagnosis. Easy. Or they can present with a lot of these congenital defects. Okay, so I do want you to go over this, right? Blue eyes, fair complexion. This is a differential diagnosis for albinism, right? Straight albinism is going to come soon, right? But straight albinism is a tyrosine problem. Tyrosine hydroxylase is not making melanin, right? But then PKU also has people not making melanin, right? Both of them are differential diagnoses of each other. One is going to have all those, major problems, have that metabolic acidosis. One isn't. One is just a skin problem, right? So you need the diagnostic screen in the newborns and fix it with replacement of the diet, right? A lot of gums, they have warnings, right? Not for phenylketonurics, right? Low the aspartame in the diet as well, the artificial sugar. Don't give them that. BH4 supplementation, and make sure that you give them tryptophan or levodopa to fix their deficiencies. Okay. Now, I found this great diagram to help you summarize a lot of these diagnoses. Okay. If they say mousy smell, it's phenylketonuria, right? Musky smell, phenylketonuria. Maple syrup, MSUD, right? Well, we're going to look at it. Cabbage smelling person, tyrosinemia, right? Rancid butter, tyrosinemia. Black urine color, alkaptonuria. That's not a smell, but black urine color. Okay. Even this, both of these are tyrosinemia type 1, right? Which means tyrosine metabolism is affected. You're accumulating a lot of it, and it might go into and make some other compounds. That's it. Okay. So it's a good sort of summary table for you. Again, let's pause it here, read the thing faster, right? And let's move forward, right? So main things that I see here: 40-year-old. A lot of the things that might affect a 60, 70, 80-year-old is happening in a 40-year-old. He also has a ruptured Achilles tendon, kidney stones, tiny black stones in his urine, right? I don't remember studying any black stones in the urine, right? Then the sclera is turning black, bluish black deposition over the pinna. Parts of his sclera are restricted, and then he has, mobility issues. So this person has alkaptonuria. Okay, alkaptonuria. You see, there's something called branched chain amino acids, BCAAs. No, that's MSUD. My bad, right? Alkaptonuria. Alkaptonuria is a part of tyrosine metabolism, where tyrosine ends up making this acid right here, homogentisic acid, right? This acid eventually goes and it makes acidoacidic or fumaric acid as it needs to, right? But this whole pathway is a part of tyrosine metabolism, right? If you have problems with this enzyme right here, okay, the processing of I think it's the dehydrogenase enzyme, right? Homogentisic dehydrogenase. This is not working anymore. You're gonna have no further processing. This is gonna accumulate because this can undergo oxidation, and if it deposits anywhere, it'll cause that black color pigmentation. Okay. Now, you may know another thing that causes black color liver, right? Not joints, not eyes, but black color liver, right? If you know the answer, share it, right? You can, or you can search for it, right? But there's a disease that leads to black liver, right? All right, moving forward. So that is alkaptonuria, right? Everything else is, just effect of that deposition. Okay. Next comes organic acidemias. Now, this essentially is a part of gluconeogenesis, right? But I put it here because the next video about glucose metabolism would be very big, right? So protein metabolism, or just gluconeogenic um, compounds, they end up going and making one common thing, which is propionate, right? Which is basically a fatty acid. Fatty acids, this propionate eventually goes and combines with coenzyme A, requires a carboxylase. We saw this, right? ABC enzyme ends up making methylmalonyl CoA, right? And eventually goes into the TCA cycle, right? By the mutase. Now, the problem is if PCC is not working or this mutase is not working, you are accumulating fatty acids, acids. What are you going to lead to? Anion gap metabolic acidosis. Energy generation is going to be low, so fasting blood glucose low. Keto acids are going to be high because every time you accumulate acids, they turn into more stable compounds, which is ketones, right? And eventually what ends up happening is, because all of this, acid and gluconeogenesis, they're not actually allowing the urea cycle to function too much. So the person starts to have hyperammonemia. Okay. So either it's PCC or it's the acidemia, right? Well, the mutase problem, right? Both of them are causing acidemia. Now, here you see two things. B7, severe B7 deficiency can lead to organic acidemia. Severe B12 deficiency can lead to organic acidemia. The difference between the two is the presence of methylmalonic acid. Okay? If methylmalonic acid is high, you have B12 deficiency. If it's low, that means you're not making it, you have biotin or PCC problem. A lot of times you'll see they, in the lab values that they give, they're going to say methylmalonic acid high. Well, you're wondering, like, where did this come from, right? But you see all the symptoms of vitamin B12 deficiencies, but then they say methylmalonic acid is high, right? That's them pointing to vitamin B12, and you should know that association, okay? Because it's not allowing you to go forward. All right. Now, you can go over this. I think I've covered most of everything. You see, because of that, liver problems and all that, you may see some sweaty feet odor, fishy odor, right? Because you're making keto acids, you're making all these like substrates and stuff. They might, show up in the sweat glands, and then you get the odor, right? Low urea cycle because of all these factors, right? Cofactors are depleted, acidosis, you get hyperammonemia, and then you see all the symptoms of hyperammonemia, which we discussed. Okay. Management, you have to decrease the amount of protein and odd chain fatty acid intake, right? You see all of these odd chain fatty acids and all of this. You see, I didn't mention it, but pay attention to the ones that are undergoing the metabolism here. Okay. So decrease the immediate consumption, make sure they're well fed so they never go into gluconeogenesis.
Real WWE back. It sounds like there's going to be like a TKO versus WWE type of.
All right, now comes MSUD, maple syrup urine disease, right? Here is where I wanted to talk about the branched chain amino acids. Okay, branched amino acids are just isoleucine, leucine, and valine. Okay, now all three of these, if you come back here, all three of these are under the control and mercy of alpha ketoglutarate from TCA cycle. They give the amino acids, the NH3 to the glutamate, and they end up making all these acids, right? Under the jurisdiction of the branched chain dehydrogenase, right? So this dehydrogenase is eventually going to make the coenzyme A, and eventually they'll make these substrates, maybe going to TCA cycle again, maybe going to fat formation, whatever. Okay, so if the muscle needs to metabolize all these three, especially these three amino acids. It has to do all of this in order to metabolize it, and then it may send it to the liver, and then you process it for energy, right? But if you're not doing it, then you get the problem, right? And they've also given you the structure in this thing. I know it's blurry, but work with it, right? And if you have the mutation, you cannot degrade the branch in amino acids, and you're accumulating things. Eventually, they make ketones, as we saw. Longer the acids sit, they end up making ketones. Hyperammonemia, because you're not transferring that NH3 too much, and because you have hyperammonemia, everything else is given, right? This is all the effect of hyperammonemia. Okay, so now you can read it. So you see branch in amino acids, N, yes. What is the main cofactor which deals with these three amino acids? It's B1. Okay, B1 deficiency will lead to MSUD, essentially, right? Because you're not gonna process your branch chain amino acids, right? So what ends up happening is, you see all these keto acids will start to increase in the blood. So if they ever say there's high amounts of alpha keto acids, and then they say vitamin B1 levels were checked and there's low, that is low, it's MSUD. And what do ketones do? Ketones smell, right? So this smells like maple syrup, burnt sugar smell. Okay, I hope that makes sense. So treatment is just the same. Restrict these three things and give them thiamine. That's it. Easy. Okay. Lastly, a couple more. Cystinuria, right? Cystinuria is excretion of amino acids, right? Especially which ones? Sorry, poor reabsorption, not excretion. Poor reabsorption of these amino acids. What ends up happening is they go into the urine and they crystallize. Okay. All of these, you see, the transporter defects leads to crystallization during acidic pH, right? So if you have acidic pH, that's when these guys crystallize. So what is the treatment going to entail? Alkalization of the urine. Important. Okay. You make the renal stones in acidic nature, but you have to dissolve them in order to dissolve them, you have to use alkalize. Okay. Potassium citrate, acetazolamide, etc. Okay. How do you test it? They've given it to you, urine sodium nitroprusside test. It's just how purple can it get. That's easy. Okay. Same thing. Any oxidizing thing is they're gonna use the one the nitroprusside test. Okay. Now in this case, you can use these two things, penicillamine, uh, thiopronin, because it can bind and sort of remove that cysteine. Okay. And the rest is sort of understood. So that is cystinuria. Okay. Now, last couple of diseases, Hartnup disease. We will, I think we'll see it again in B3 deficiency, right? But this is basically B3 deficiencies, which leads to amino aciduria. That's the main thing, right? Because you have this tryptophan, and tryptophan is not making, not going further and making a lot of these, proteins and stuff, right? You end up getting the B3 deficiency and all the tryptophan, like serotonin problem, neurological problem, etc. Okay. Transport of neutral amino acid defect, all of that is going into the urine, right? And it makes this, smelly poops, right? How do you fix it? Increase the amino acid, sorry, give them proper protein diet and niacin supplements. It should fix itself, right? But usually all these neutral amino acids, they're being lost in the urine. Okay. Albinism, we kind of saw it already, but I've just placed it here again because this is all, tyrosine based again. So phenylalanine hydroxylase not making tyrosine, right? Tyrosine not making melanin. You see, copper is also mentioned here. Important. Okay. They've given you the pigmented skin disorders. I've added them here for your differential, right? There are a couple more differentials, but this will definitely be, your top four, right? If it's to do with inborn errors of metabolism, I've given you this high yield table: albinism, you see, Chirek Higashi, which is an immunodeficiency problem, and then phenylketonuria, which we discussed, right? So if they ever say, albinism or patchy, hypopigmented skin, this is what you're looking for. I think we understand most of these, maybe not the mechanism, but at least, I think this one is the one that sort of stands out. See, patchy depigmentation of skin, hair, and irises that can be associated with deafness, right? Neural crest cells have not actually, invaded the skin. Okay. So finally, this is your high yield disorders of amino acid metabolism. I think we covered mostly all of this. And your differential is ready. Okay. You've understood everything about proteins. Moving forward, we're going to do glucose, glycogen, all of that, right? And we're going to be done with metabolism. All right, then. Take care.